Okay, good afternoon. We will start. I have a couple of things to just bring to your attention. Next week is the last week of the course in this session, and the topic is the one that was snowed out earlier in the year. So you'll see on the website the details about the speakers and the topic, which is a very important one. It's a combination of the issue of screening for malignant disease, both genomically and looking for markers, and then the case history is that of prostate cancer, which was presented. And then the other speaker has analyzed this from the standpoint of kind of, are we doing the right thing? And where do you draw the line? And it's really an extraordinary, provocative issue, which is very much at the forefront now of new technologies and their application. So that's next week. The final exam, a backbreaker of a computerized exam, which has been passed with high grades by everyone who's taken it. So those of you who haven't and want to receive a certificate for attending these course, you can pick it up online on our website. Okay? So today's subject of malaria, and particularly our two speakers, whom I will tell you briefly about, is particularly appropriate for the sort of logo of the Brooklyn Bridge, which just disappeared. Those two gentlemen who are on the ramparts are discussing. One is saying, what's the origin of malaria? Where did Plasmodia come from? Are they 50,000? Is it 50,000 years old or 100,000 years old? And what's the relationship? Why do some people in some parts of the world seem to be resistant and others aren't? What can we do about it? It's a huge problem, because even then they knew that this was a global problem. This is the end of the 19th century. And of course, today we know that there are several 100 million people who suffer from malaria, with a fairly high mortality rate, particularly amongst young children, largely in sub-Saharan Africa, but elsewhere in the tropical and subtropical area of the world. And the other chap is saying, but look, this thing is transmitted by a mosquito, the Anopheles. Maybe we can understand what's the interaction between the parasite and the primary vector, the mosquito. And if we understand something about what happens in the mosquito, maybe that will give us some clues as to how we might even be able to target some forms of therapy. Because in the last analysis, back then and even now, therapy has been a two-edged sword. There is much public health ventures to deal with water, with netting over bed at night, because it's the female Anopheles that does the eating of human blood, and basically then picks up the one form of the parasite, lets it multiply, and then delivers it to the next victim. There are drugs which are effective, and in some of the more virulent forms of malaria have undoubtedly been life-saving. But there are huge problems, and there's drug resistance and a need to develop perhaps new agents if you knew what you were targeting. And then behind all of this is an incredible story of world history, warfare, poverty. The effect of this disease has been probably much greater on the history of the world than many of the things that we temporarily think are so important. Well, So our two speakers today are Carolina Barillas-Mury, who is here at NIH. She is originally from Guatemala, where she graduated and in medicine, and then went to the University of Arizona, where she received a PhD degree, and then was a postdoctoral fellow with a very famous professor at Harvard, Fotis Kafatos, who oscillated between Cambridge and the European Molecular Biology Lab in Heidelberg, and that's where she received her postdoctoral training. And then she went to the University of Colorado State University for a while, came here in 2003 in NIAID, and is now a senior investigator in the Laboratory of Malaria and Vector Research, and the head of the Mosquito Immunity and Vector Competence Section. Carolina is highly honored. Next one, please. And most recently, actually, there's a mistake in this slide because it wasn't yesterday, it was today that she was elected to the National Academy of Sciences. Her studies deal with the interaction between the Plasmodium, the parasite, the gut microbiota, and the epithelial cells of the mosquito. Her focus has been pretty much on the mosquito. And so we all congratulate you and look forward to your presentation. So next one, can you go back one, please? No, keep going. Yeah, okay. So our second speaker, who is a guest visiting from the University of Pennsylvania, is Beatrice Hahn, where she's professor of medicine and microbiology. She received her medical degree in Munich and then came to the NIH as a postdoctoral fellow in Bob Gallo's laboratory, where she became acquainted with and learned retrovirology, which has been the basis of her research. Initially at the University of Alabama in Birmingham, where she was for a relatively long time, and then 3 years ago moved to the University of Pennsylvania. Beatrice is also a member of the National Academy. Today is Double Threat Day. I mean, we really have two superstars. And her main interest has been in looking at the origin and the evolutionary implications of the origin of initially HIV, particularly simian retrovirus, SIV, and then in recent years has turned her attention to malaria. And her studies, which are on the website, I think you'll find extraordinarily exciting and interesting, and I will not belabor that any further. So I think, Beatrice, you're going to be the first speaker, right?
Yes, thank
okay.
you. And
welcome.
That was a very nice introduction. Does this work? Yes.
Oh, by the way, that's her in the slide up on the top.
So I need the, yes, thank you. So I will tell you a little bit today about the origins of two human malarial parasites, Plasmodium falciparum and Plasmodium vivax. This is sort of an outline of my talk. I'm going to give a very brief introduction, then talk about Plasmodium infections in African apes, then cover the origins of the two parasites, talk a little bit about the likelihood of additional Plasmodium zoonoses, and then speculate a little bit about the age of human malaria. So just to bring everybody on the same page, because I heard that you have very different backgrounds, you must know that malaria is caused by a parasite of the genus Plasmodium. There are actually 5 Plasmodium parasite species that infect humans. They're listed here: Plasmodium falciparum, vivax, ovale, malaria, and nosei. And it's really falciparum in particular, and also ovale, that are the major killers. Falciparum being ravaging through Africa, and vivax being the most widespread malarial parasite on the globe. As Wynn said, they are being transmitted by mosquitoes of the Anopheles genus. Size matters. Well, Wynn mentioned that I'm an HIV virologist, really, by training. And so going from 10,000 base pairs to 14 chromosomes is a big jump. So we had to get used to it. So falciparum has 14 chromosomes, about 5,300 genes. Human genome is about 100-fold larger. Bacteria are smaller, and HIV is very small. And we still haven't figured that one out, so that tells you how long we're going to work on this. So what does the extra genome size buy you? For starters, a complex life cycle. And you've all seen the schemes of the life cycle. There is the human or ape host, and then there is the mosquito host. And you know that I'm not going to go through the details because it doesn't matter for my talk, and I'm sure that Carolina will cover the mosquito stage. The important point is there's a liver stage and there's a blood stage, and then there are gametocytes that are being picked up by the mosquito again. And this is where sexual reproduction occurs, and this is where asexual reproduction occurs. The point is malaria is a bad thing. Almost half of the world's population is at risk. There are 300, 500 million new infections every year, over 1 million deaths, primarily in kids in Africa. It's something you don't want to have. There's no good vaccine yet, although it's looking more promising, and there's widespread drug resistance, so this is a problem. So phylogenetically, in the evolution, where do the human malaria parasites fall? This is a phylogenetic tree of sequences from a variety of different plasmodial parasites. And you see here there are 4 groups. There's a primate group 1, a primate group 2. In the group 2 is falciparum. In the primate group 1 are the other 4, vivax, nosai, malaria, and ovale. And they are interspaced with parasites from rodents and from birds and reptiles. So this is important because this lineage here is quite divergent from the other lineage that includes the human parasites, and actually has been some time ago was proposed to be named a different subgenus, Laverania, and that you will find that that actually is a very helpful nomenclature as I go on. Here we go. So there are a lot of parasites infecting primates in Asia, macaques, gibbons, including orangutans. There are also Plasmodium parasites in the New World, in the Americas. However, it should be pointed out that these are actually reverse zoonoses. There are a number of New World monkey species infected, which is called Plasmodium brasilianum, but it's actually P. malariae that was brought to the New World and then jumped from humans to monkeys. Similarly, Plasmodium simian infects howler monkeys and spider monkeys, and this really was brought to the New World by humans, and it's P. vivax. And it's important because it shows you that these parasites can jump species. In Africa, there are parasites in monkeys, but also in apes. P. raikanaui is well-known chimpanzee parasite, but about 100 years ago, people were studying Plasmodium infections in apes in Africa, and at the time they didn't have any genetic tools, but they looked in the microscope and found the same thing that they could find in humans, and so they gave them names, Plasmodium schwetzii and rhodani, and unfortunately there are no stored samples that would allow us today to figure out what they actually were. And then there is Madagascar and the lemurs. They have their own species. So one thing that's important that I want you to take home is that these 2 groups of Plasmodium parasites are very different with respect to their host species specificity. These guys here are quite promiscuous. I told you about the human parasites going into New World monkeys. But as it turns out, that Plasmodium nosai is a true zoonosis. That is a parasite that infects macaques in Southeast Asia, and that jumps over to humans and causes quite a bit of mortality, morbidity, and even some mortality in these areas. And it's, I think, still not shown that this parasite can be transmitted from humans to humans. In each case, it is a macaque to human transmission. Cynomolgi and Inui also have been found in humans, although in these cases, these are laboratory transmission. But still, it shows you that this group, what we call non-Laverania human or primate parasites, is quite promiscuous. This is not the case with falciparum and Reichenau, because about 100 years ago, people who were studying ape malarias in Africa were doing transmission experiments, something that is unheard of today. But they showed that while they could transmit some of these from apes to humans and humans to apes, they could not do that with parasites infecting chimpanzees. So given this phylogenetic relationships, somehow my punchline was stolen here. The animation doesn't work, but it's okay. So what people had actually thought of coming from this phylogenetic relationship is that they saw a chimpanzee parasite and a human parasite, and they said, well, maybe these parasites have co-evolved with their host, and hence the split was when humans and chimps split about 6 million years ago, and so most likely these parasites have been in these species for this long a period of time. And because Cynomolgi was the closest relative to P. vivax, and because there are a lot of other macaque parasites here that fall in this same group, people said, well, vivax must have originated in Asia, and it must have been a cross-species transmission from an Asian monkey parasite. And both of these hypotheses are wrong, and I'm going to show you why. And if there's nothing else that you take home today, take that home. Vivax is not of Asian origin, and Plasmodium falciparum did not co-evolve with its host. Why do I know this? And how does an HIV person get into the malaria field? The reason is we've been interested in the origin of the human AIDS virus, HIV-1, and we traced it back to chimpanzees. And the way we did this is non-invasively, because chimpanzees are highly endangered. You cannot study them in the wild by anesthetizing them or doing anything invasive to them. So the only thing left for you to study are fecal samples. And so we've been doing this for the past decade, collecting fecal samples from chimpanzees, bonobos, gorillas, and it is amazing what you can get out of fecal samples. For starters, you can get antibodies. You can do a Western blot. If you have an infection, you can do a more sensitive type of Western blot, and you can find antibody reactivity. But you can also extract nucleic acids, which allows you to extract viral RNA in the case of SIV, or host DNA. And so you can get full-length and partial SIV genomes. You can do phylogenetic analysis. You can even reconstruct infectious molecular clones. You can resurrect the virus from fecal samples, which was done in the case of the gorilla virus, for which there is no isolate. But you can also study the host. You can do mitochondrial DNA analysis, and you can verify the species and subspecies. And you can enumerate individuals, which allows you to do prevalence determinations. So we've been doing this for HIV, and we actually traced it back to the pandemic form of HIV-1 to chimpanzees, and the non-pandemic forms to chimps and gorillas. But, and we did this back in 2006. But in 2009, a paper was published by a French group, in fact, working with our collaborators, Martine Peters and Eric Delaporte. And what they showed is that you could also detect Plasmodium sequences in fecal DNA. And at that point, we knew we were in business because we had 10,000 in the freezer. And so it was a no-brainer to look of what we could find in these samples. Incidentally, when I was first approached to look for that, I told the person, this was actually Richard Carter, who has been in the malaria field for a long time, I said, you will never find Plasmodium DNA in fecal samples. Why would it be there? Turns out I was dead wrong, and the reason it is there is because of the liver stage, and we just published a paper, or it's in press, with Richard Cullerton, showing that a liver infection actually leads to secretion of parasite nucleic acids into the bile and from the bile into the fecal sample. And that's why you get Plasmodium sequences in fecal samples. But this took some time to figure out. So given that, we wondered what might we find in apes. And we knew from the old studies that there were equivalents of all of the human parasites in apes. So Reichenauii was the chimp equivalent of falciparum. Rodani was the chimp, or people primarily looked in chimpanzees, but some did also in gorillas. So Rodani was the equivalent of malaria, and because vivax and ovalia are difficult to differentiate, the equivalent was Schwetzi. But only Reichenauii made it into the modern day in terms of culturing the parasite and doing the genetic analysis. No samples were left from these descriptions, which were basically a description of blood smears and the morphological analysis done back by Edward Reichenauii in Cameroon and these other people in Sierra Leone 100 years ago. They were interested in human malaria, and they figured, well, maybe our closest relatives have them too, and sure enough, they did. So the questions we wanted to address, given our repository, we wanted to know which ape species and subspecies are actually infected in the wild with Plasmodium. How prevalent was this infection? How many different Plasmodium species were circulating? And were the human parasites of ape origin? And so we did what everybody else did, is we targeted a region of the Plasmodium mitochondrial genome, amplified about 1,000 bases. This is in the CytoB gene, and simply took our fecal samples, extracted DNA, and screened the hell out of them. And the results were, we found ape Laverania infections all over the place. Every single chimpanzee subspecies, of which there are 4, the Western chimp, the Central chimp, the Nigerian Cameroonian chimp, and the Eastern chimp. Every single subspecies was infected, and you can see here the field sites where we collected samples, in yellow being those that were actually positive. Now, interestingly enough, chimpanzee, there is a second species of chimpanzee, that's the bonobo or the pygmy chimp. We did not find any infections in these animals. The range of the bonobo is south of the Congo River. And I show 4 field sites here, but we have since expanded into 8, and we still haven't found it. Gorillas, similarly, this is the Western gorilla, this is the Eastern gorilla. Lots of infections, highly prevalent in Western gorillas. We did not find any infections in Eastern gorillas, but I have to say, we only looked at 146 fecal samples there, whereas we had a lot more, close to 3,000 fecal samples here. The reason being, our man who collects from these populations said there are rebels in the forest with machine guns, and I said, don't go there. So we only have few samples. So hopefully when the political situation in the DRC calms down, we will get more samples from that region. So chimpanzees and Western gorillas infected at a high prevalence, and remember, this is a fecal prevalence, so it's probably a vast underestimate of what is actually there. We then put these sequences into a phylogenetic tree, and we had a surprise. Instead of the 2 lineages that were known before, Reichenauii here, and this is human falciparum, we actually found 6 major lineages, which translate into 6 species. 3 of them in red were specific for chimpanzees. 3 of them in green were specific for gorillas. And you see here where human falciparum falls, it falls within the radiation of one of these 3 gorilla parasites. And every single human sequence coalesces back to one common ancestor, and that ancestor falls within the radiation of the gorilla parasites. And that told us there was a gorilla to human transmission that generated human P. falciparum. So this is now in schematic form what we found, and we've since given these lineages, species a name. Some people object to that because giving a species name requires more. It requires morphology and other things, but because wild apes are inaccessible, this is all we can do. And the reason we have given these species names is so we can refer to them. So we can differentiate what we call P. gamoni from chimpanzees from P. reichenowi. And remember, the topology is always the same. P. reichenowi is the closest relative of what we call P. pre-falciparum, because it's the gorilla parasite that gave rise to falciparum. And P. gamoni is always the closest relative of P. what we call P. adleri. And the other two fall in the middle. And here are the non-Laverania parasites. So take-home points at this point. Chimps and western gorillas are each naturally infected with 3 Laverania species. Ape infections are highly prevalent, widely distributed, and almost always comprised of mixed parasite infections. I didn't show you that, but we would almost always find not just one, but all 3 of the chimp or gorilla-specific parasites, but never the cross-species transmission. And they are remarkably host-specific, so you must understand gorillas and chimps live in the same forest. They eat in the same fruit trees. They will be likely bitten by the same mosquitoes, yet they have very species-specific Plasmodium parasites. Humans acquired falciparum following a single gorilla-to-human transmission event. Falciparum parasites in captive bonobos and chimpanzees are of very recent human origin. I didn't show you this data. People, of course, first looked in captivity, what they could find. And they would find human P. falciparum, and they said, oh, bonobos are the origin of human P. falciparum. But it so happened that these bonobos had falciparum parasites with human drug resistance mutations in them, and they were in an urban sanctuary. So what it tells you is bonobos, although they are not infected in the wild, they certainly are susceptible to infection. If you put them in a captive setting and you put the right mosquitoes around them, they will become infected. Why they're not infected in the wild is still a mystery. And I also didn't show you this data, but there is no evidence of human falciparum in wild-living apes or monkeys, and we've looked. So what about the other Plasmodium species? Well, when we did the survey, we found also evidence of ape relatives of ovale, vivax, and malaria. And in fact, there are other lineages here that I am not showing here that would indicate that apes have additional Plasmodium species that do not have a human counterpart. Now, because of this, finding vivax in apes, that was of interest to us. And it was of interest to us because of the epidemiology of vivax compared to falciparum. So this is the global spread of falciparum, and this is the global spread of vivax, and it's almost superimposable with one big exception, and that's here. There is no vivax in West Central Africa, whereas there's plenty of falciparum. And why is that? Because people here are Duffy negative. They don't have the Duffy antigen, which is the receptor for P. vivax. And people have always wondered, why is this? Where is the pressure of people to have developed this genetic mutation? So this is the Duffy antigen receptor of chemokines, or DARC. It is a chemokine receptor, normally expressed on erythrocytes and other tissues. It happens to be the receptor for vivax and nosy. It interacts with the Duffy binding protein. That's the ligand on the parasite that interacts with this receptor. And if you don't have the receptor, you generally do not get infected. This was discovered here. Lou Miller actually discovered this. And in humans, humans have really a fabulous mutation. It's a mutation in a cis-regulatory domain of this protein that abrogates expression, not in general, but just on erythrocytes where it counts. So we knew human P. vivax was virtually absent from West Central Africa, yet there was a mystery, and that mystery has been observed for quite a number of decades. A certain percentage of travelers that return to Europe or to the United States or to Australia that have visited these parts of Africa come back with malaria, and when you look, it's not falciparum, but it's vivax. And people say, well, where did these people pick up the vivax? There is no vivax there. And you never know, some of the people travel to other places as well. But consistently, and there have been papers written about these unusual cases of travelers bringing back vivax. In addition, Richard Culloden published a paper some time ago where he looked in the blood of inhabitants of a city in the Republic of Congo and found antibodies to vivax. So how did these people get antibodies to vivax if it wasn't there? So we were wondering whether there is a natural reservoir, obviously, in apes. And so we did the same thing as before. We targeted the Plasmodium mitochondrial DNA. This time we didn't go for the CytoB gene, but for the COX-1 gene because of primer design and other things. And we screened again, went back to the freezers, pulled out the fecal samples, extracted DNA, and screened again. This time we screened about 5,000 fecal samples. And here are the results. We found definitely vivax-like parasites in central chimps, eastern chimps, western gorillas, eastern gorillas. We didn't find it in bonobos again. And we didn't find it in one subspecies of the Nigerian Cameroonian chimp. But the problem here is vivax does not grow to the same titers in the blood as does falciparum. And so it ends up in the fecal sample in lower amounts. And so we had to screen on average 100 fecal samples to find one positive. That's a lot of fecal samples to go through. And it's clearly a titer business. So we got infection rates, which were much lower than what we had seen for the Laverania. Laverania were 40 to 50%. Here we're talking 4 to 8%. But even in humans, parasite rates of P. vivax in endemically infected populations hardly ever exceed 7%. So we're clearly in the transmissible range with these numbers. And in fact, in humans, a parasite rate of greater than 1% indicates stable transmission. So this is, again, the map. Again, yellow means P. vivax was present. So you see the spread. Again, there are a number of negative sites, but I think that's because we missed it, because we didn't have enough samples to screen. And we even found it in the Ugala region of Western Tanzania. This is a very unusual region where there are chimpanzees that are called savanna chimps. They are different from their forest counterparts because they have such tough environmental conditions to deal with. 6 months out of the year it doesn't rain. They are behaviorally different from their forest counterparts. And even there, we found P. vivax not just once, but 4 times. So it's a widespread. Again, nothing in bonobos, and I don't know why. So at that point, we wanted to do the same thing, amplify genes from the mitochondrial genome, from the apicoplast genome, from the nuclear genome, to compare the ape versions to the human versions in order to figure out what the origin was. And because of the low parasite levels, although we started out with the diagnostic fragment in the mitochondrial genome, we had about 80-some positive samples. But when we tried to amplify larger pieces, which would be necessary to have a decent phylogenetic analysis, it got pretty slim here. And so we realized we had to do something different. And so we went and looked for African sanctuaries. And African sanctuaries are great because these are the places where the bushmeat orphans end up. So the parents of these chimps were slaughtered for bushmeat, and they end up in these sanctuaries. They are in captivity. There is a fence fencing them in, but the habitat is immediately adjacent to the habitat of wild chimps. So they get bitten by the same mosquitoes. Yet you can treat them if they're sick. You can do the annual health exam. You can draw blood from them. And in fact, some of them, the reason we even got involved with the sanctuary is because the veterinarian came to us and said, can you see whether some of our chimps have malaria? Because they seem to have malaria and they have the symptomatology of malaria. And when we give them antimalarial drugs, they get better. So that's why we started to screen these apes. And that's why we had access to blood samples. And so when we did this, this is what we found. This is the mitochondrial genome. It's a large fragment. It's about 3 kb in length. The sequences are color-coded. Here are the central, Nigerian, eastern chimps, western gorillas. So you see a phylogenetic tree where the human P. vivax strains, and they come from all over the globe, again form a single lineage that coalesces to a single common ancestor, which falls within the radiation of the ape parasites. And this is true for the mitochondrial genome. It's true for a nuclear gene, LDH, same thing. And it's true for an apicoplast gene, CLPC. Here there are fewer sequences available, but same story. All the human sequences formed a single lineage that fell within the ape radiation. And when we looked at the diversity, comparing human vivax and ape vivax, we always saw that the ape parasites were more diverse, which is what you would expect from a reservoir. At all loci tested, it differed from 1.4 times more in the mitochondrial gene to up to 50 in some intergenic regions. And so clearly the reduced diversity in human vivax reflects a recent bottleneck. Now, given that vivax is from the more promiscuous lineage, we did want to be sure we didn't miss something in the monkeys. So we did look at 1,000 blood samples from primate bushmeat samples. These are monkeys that are sold at bushmeat market for meat. And our collaborator, Martine Peters, has drawn these blood samples. They are from way back when we were studying SIV, so these samples came handy. Again, we looked at 16 different species at 11 different locations at these sites, and we found no evidence for P. vivax. But half of the samples had hepatocystis, which is a known monkey parasite. We also looked at the DARC gene, the promoter region, and the gene itself, because we wanted to know whether the bonobos had a Duffy negative genetic mutation. You can't see it, but this is the Duffy mutation, but all the apes were wild type. None of them looked like the Duffy negative mutation. And then there is an amino acid here at position 42, which is associated with a potentially protective allele in humans. And again, all the apes were wild type. Now, they had other changes, so I can't exclude the possibility there's a Duffy-like change, but at least the human positions were wild type. So I've shown you this now. This is human falciparum within the radiation of ape Laverania species. Here's human P. vivax within the radiation of vivax-like parasites in apes. So what would you conclude about the origin of human P. vivax? Would you say this is a single ape to human transmission? No. Why not? Because of this. No species specificity between the chimps and the gorillas. Strict species specificity. So clearly the parasite goes from gorillas to chimps, from chimps to gorillas, and most likely at some point in the past went from gorillas to chimps to humans to gorillas to chimps. In other words, there was an ancient stock of vivax that was promiscuous that went around all these different species. And then the humans developed the Duffy negative phenotype and basically eradicated vivax in the human population. And what you're looking at here is one parasite that escaped out of Africa and now is circulating in global populations. So, origin of P. vivax, P. vivax does not divide into chimp and gorilla-specific lineages. This is the ancestral stock I just talked about that was probably present in all ape species until the Duffy negative mutation arose and started to spread. And thus, extant human P. vivax represents a bottlenecked lineage that survived after spreading out of Africa. Take-home points, chimps and gorillas, but not bonobos, are a substantial P. vivax reservoir. There is no evidence of P. vivax-like infections in other African primates. There is no evidence of a Duffy negative phenotype or selection of a protective allele in wild apes, although I cannot exclude that there are other mutations that could change the pathogenicity. Human P. vivax arose from within a Plasmodium species that infected humans, chimps, and gorillas. And this provides a long-sought explanation for the selection of the Duffy negative phenotype in West Central Africa. There was the reservoir, there was the transmission, there was the reason to develop a Duffy negative phenotype. So since we had now, the whole Laverania subgenus with 6 different ape lineages and human and ape P. vivax, Sish in the laboratory asked, does cross-species transmission occur in areas where infected apes and humans live in close proximity? What about other zoonoses? And so we went back to Cameroon with our collaborator who had not just collected blood samples from primate bushmeat samples, but also from humans, and especially humans that would live in villages close to the natural range of both chimps and gorillas with high prevalence of Laverania infections. And so we screened those people. And so the first thing we asked, and this is a view of one of these villages, which shows you that people truly do live and sleep within mosquito flying distance. So the first question we ask is, most of falciparum sequences at this point had come from isolates, not necessarily from West Central Africa. So we said, well, maybe we missed an additional transmission, because in HIV, there are 4 transmission, HIV-1 groups M, N, O, and P. And we said, well, maybe this was also going on and we just missed it. So we looked at almost 250 new infections, but every single one fell within that lineage of human P. falciparum. There was no evidence of any additional cross-species transmission, which is odd because once something jumps, it works. It should jump more than once, unless there's something really unusual going on. We also did some pyrosequencing because we thought maybe people are infected with human falciparum. They also get infected with the ape versions, but the ape versions aren't adapted to humans, and hence their titers are lower, and we missed it. So we did deep sequencing to see whether we would find it. And so in almost 450 human samples, we looked, and all we would find is either a monoinfection with human falciparum or various combinations of falciparum, ovale, and malaria. We did not find any ape. Yet, when we looked in ape fecal samples with the same primers, we did find all of these different species. So it wasn't that the primers weren't working. It was that these people were not infected with ape Laverania parasites. Yet, the same group who discovered that you could amplify Plasmodium sequences from fecal samples also looked at vivax, and they did the same thing we did, except they didn't do it in thousands of samples. They went to captive chimps and gorillas. So here's a chimp, here's a gorilla. They were in sanctuaries and zoos, and they found ape vivax in them. Then they looked in Anopheles muceti from these regions. They put out traps close to where the apes were and caught over the period of a year a couple of different positive Anopheles, and showed that they were infected with the ape parasite. And then they found one traveler, one French forester who had worked in a forest in the Central African Republic for 2 weeks, returned back to France, had malaria, looked at it, looked like vivax. But when the sequence was determined, it was an ape version, not the extant human version. And in fact, because we had so many more ape sequences, we took that sequence and put it into our tree. And here's the extant human vivax, and here's the traveler. So clearly, the traveler had picked up an ape parasite. So bottom line, in West Central Africa, people are protected. They don't have Duffy. They don't get infected. You bring in a Duffy positive, and it goes to the right place, gets bitten by the right mosquito, comes back with an ape infection. So ape vivax clearly has the potential to cause zoonotic infections. And this is 100% in line with what people have described 100 years ago about experimental transmissions of, at the time, P. schwetzi from apes to humans, which worked, and yet trying Reichenau from chimps to humans failed. So this is exactly what you would expect. So take-home points at this point: wild apes are infected with Laverania parasites that do not serve as a recurring source of human malaria. And so it really leaves us hanging there with an explanation why falciparum, falciparum from gorillas crossed to humans in the first place. And maybe Carolina will have an answer one of these days. She certainly had an idea. Humans are susceptible to infection with ape P. vivax. And so the question is, what would happen if you bring in an influx of Duffy positive people into areas where ape P. vivax is endemic, and then perhaps export that back out to places in Asia where vivax is normally transmitted? Ape vivax is more diverse and perhaps a bit more versatile. So this is something to watch. So how old is human malaria? And there I have to speculate. So there are two schools of thought. Some people think it's very ancient, hundreds of thousands of years old. Other people think it's very recent, less than 10,000 years old. The people that think malaria in humans is ancient base their conclusions on molecular clocks, and also assumptions that certain parasites have co-evolved with their hosts. And then they use evolutionary rates, not from Plasmodium, because that's not known, but from other species, bacteria, to try to calculate that. People who don't believe that malaria is ancient base it on the parasite biology. They have observed that some of the genetic mutations in humans have not yet spread to fixation, that the glucose 6-PD is less than 10,000 years old, and that Duffy emerged around 30,000 years ago. So that is not compatible with hundreds of thousands of years of infection. And also, there's data that Anopheles gambiae really had a major speciation event about, I think, 5,000 to 10,000 years ago, when human hunter-gatherers got together into villages and there were pools of water and stuff like that. So if you believe the first group of people, then you just simply say, in this case, it's Reichenauii and the precursor of falciparum in chimps and gorillas, so roughly 6 million years. If you assume that, then you come out with the last common ancestor of falciparum in the 250 to 300,000 years ago. But you can also calculate this differently based on the mutation rate of Plasmodium, which of course has to be evaluated in vitro, and the number of replication cycles within a life cycle. And so these are some back of the envelope calculations. You have to account for mosquito, for liver and blood, but probably there are about, about 500 replications per year, probably not more, possibly less. And the mutation rate in vitro has been published anywhere from 1 times 10 to the minus 9 to 10 times 10 to the minus 9. And when you plug all that in, you come up with an estimate that the last common ancestor of P. falciparum could have easily existed 4,800 years ago. And that just, don't take that number too seriously. It just means that this would be more consistent with a more recent origin of human malaria. So the diversity among extant P. falciparum strains can easily be explained by descendants from an ancestor that existed within the last 10,000 years. Vivax is more diverse than falciparum, so it would be older. But because of the hypnozoid stage, this is the dormant liver infection, the number of genome replications is difficult to estimate for Vivax. And so maybe the age of the Duffy mutation, which has been estimated anywhere from 10 to 30,000 years ago, is probably a better calibration point of the time when that last common ancestor of cosmopolitan P. vivax emerged out of Africa. And to us, it seems that using mutation rate seems more credible than molecular clock estimates, which are notorious for giving you wrong results, especially in HIV when we try to do the same thing. Because the further you go back in time, the less accurate these calculations are. So future studies, very briefly, we really would like to know what happened and why the gorilla precursor of P. falciparum managed to overcome this barrier, and we do not have an answer yet. But it is known that the merozoid, which is the form of the parasite that infects erythrocytes, has many surface receptors that interact with ligands on the erythrocytes, and they are not absolutely required, but one has been recently shown by the Sanger group to be absolutely essential, and that's called, the receptor is called Basigen, and it interacts with the RH5 protein on the parasites. And so because this is such an essential interaction, we were interested in what the RH5 equivalents of our ape parasites would look like. And so we amplified the equivalents from fecal DNA, because when you target something, you can actually get it out. And interestingly enough, this is what we got. And this is what I showed you before. And what you can see here is that the order of the parasites is not right. Reichenau should be with pre-falciparum falciparum. Reichenau is here. No falciparum pre-falciparum. It sits down here. So what that suggests to us is a horizontal gene transfer that went from what we call Plasmodium adleri, another gorilla parasite, into the precursor of human falciparum. And we actually now have genomic sequences that would map this whole region to an 8 kb piece. And it's the only piece in the genome where we find evidence for this. So has it to do with the transmission? We don't know, but it surely is odd that the one thing that's absolutely essential also is involved in this horizontal gene transfer. So how can we get at the genomes of these parasites? The blood we get from the sanctuary apes, these are asymptomatic individuals. By and large, the parasite load is very low. We cannot do any preparative work there, which means we have whole blood, usually on filter paper. That's what we have to work with. And that usually doesn't work in whole genome sequencing. So Seish Sudaraman in my lab came up with what he calls selective whole genome amplification. And that is based on the fact that each species has unique mutational biases. And so you can design primers that are specific for Plasmodium and don't amplify chimp DNA. And you can then use a Phi29 polymerase that basically specifically or almost specifically amplifies your Plasmodium and leaves the chimp contaminating DNA alone. And so we've done that, and we've seen a 30,000-fold enrichment. And this is just to show that we can do it. This is chromosome 9 of P. reichenauii. It works great with blood samples. It doesn't work so great with fecal samples. So there is still some developmental work that we need to do. We even get, this is the end of the chromosome, which is usually difficult to sequence. And we got it there. And we have very few gaps in the sequence. So we will have a way to look at the genomes of these parasites and to see, because they have to have the blueprint of what happened, to see why pre-falciparum jumped into human only once. So my conclusions: 3 major human pathogens originated in apes: HIV-1, falciparum, and vivax. Obviously, the transmission hurdle for ape pathogen is lower. That's not too surprising because these parasites and viruses and pathogens have to interact with host proteins, and the closer they are, the easier it is to jump. Still, only a small subset has actually successfully colonized humans, and that tells you that there are very potent restriction factors and barriers to cross-species transmission. And understanding them is very important because it could give you a glimpse of what to do to counteract these infections. Obviously, I hope I convinced you that non-invasive studies of wild apes have been important, and that there actually is value of studying ape precursors of human pathogens in their natural hosts, although it doesn't immediately give you clinical benefit for humans. These are all my collaborators, lots of people in my lab. I mentioned Martine Peters and her team in Cameroon, my friend Paul Sharp and his team in Edinburgh, who does the phylogenetic analysis for us, lots of people who gave us human, and even more people who gave us fecal samples, and of course, our study subjects. That's it.
Thank you. Thank you very much. That was very exciting. Well, we have time for some questions, and please, I'd only ask that you either speak loudly or use the microphone so people watching can hear the question. Well, let me lead off. Why is there no falciparum in South America?
There is. There is plenty.
What?
There is plenty of falciparum in South America.
Not on your slide.
No, there was. There was.
I didn't see any blue there.
No, there is.
I thought there was. That's why
No, there is. The only big hole is here. Sorry. So, I mean.
Oh, wait a minute.
So.
Oh, yes.
So here is the vivax, and here's the falciparum.
I stand correct. Okay.
So how do you think the Laverania evolved in the apes? Did it co-evolve? The different, the C1, C2, C1, G1, did they co-evolve with the chimp and the gorilla evolution?
that's one potential explanation, but I have no evidence to either confirm or refute it. You could explain it that way. How these parasites got into the apes, I really don't know. This is also true for the vivax precursors. I don't want to make any assumptions because then I'm back to, falciparum co-evolved. But what is more interesting is why there is the strict species specificity when they live in the same place and get bitten, presumably by the same mosquito. I think one big aspect, a big whole gap in knowledge right now is the mosquito part, because we need to figure out which species are transmitting these parasites among apes and how they work, because that could explain a number of these things. Oh, sorry.
So thank you. So if you think there was a horizontal gene transfer, would you hypothesize that that would have occurred in a mosquito
I have
or in an ape?
No idea how this works. There actually is no mechanism that I could point to that would make sense. I just don't know.
Thank you.
For a long time, there was a notion that sickle cell disease has some negative pressure for transmission of falciparum. Where is that story stands today?
I think sickle cell disease is partially protective of the pathogenic consequences of infection. I don't know whether it has anything to do with transmission. I don't know. I see.
Sorry, my understanding is that sickle cell anemia protects from the severe forms, so protects children from cellular malaria, but you still get infected,
Right.
but not is not lethal. Let's say.
So is that also true for the thalassemia traits and same type of mechanism?
Yes.
So that's evolutionarily preserved essentially by reducing.
Yes, I think with those, if you are homozygotes, this is really bad because you're sick, but if you're heterozygote, then you're healthy and you're protected from severe malaria. So they have stayed in the population as in Africa. So it's another evidence of strong selection of human populations by Plasmodium.
And the Duffy, there you want to be homozygous because you happen not to need the Duffy antigen on your erythrocytes. But that is a very unusual mutation.
Are primates ill when they have infection?
I think so, sometimes. Because the reason we got into this is because the sanctuary veterinarian would describe cases that would look like cerebral malaria. And then, of course, because of this is up in the forest, there's no infrastructure, you can't even do blood smears. But when she gave anti-malarial drugs, chimps would get better. This happened 3 or 4 times. That's why she came to us and said, tell me what's going on in my apes. Now, we see a lot more infections than we see disease. But that could be a similar story with humans, where most people have immune response that is partially protective. And it only hits the kids and it hits the pregnant women.
But in humans, it kills infants. What about in the primates?
We don't really know, because these apes are 5, 6, 7 years old.
You touched on it a little bit. Do apes also suffer from pregnancy-associated malaria infections?
Is what?
Do apes also suffer from pregnancy-associated
I do
malaria
not know,
infections?
because one thing they don't do in the sanctuaries, they don't breed them. They don't need more. They can hardly feed the ones they have.
Thanks for your presentation. I would like to know what the level of drug resistance is in apes as compared to humans.
We haven't looked yet. We're just beginning to get the whole genome to look at the genes that are involved in that. We haven't looked yet.
Thank you.
I would expect none.
Is there other ways of transmitting the virus rather than mosquitoes? For instance, from the mother to the fetus during pregnancy, or
In apes, I have no idea, but I think I would speculate that ape, Laverania, and even vivax infections are very similar to their human counterparts in these things. But I have no knowledge what happens in apes.
Do you know, are there mutations that affect either the capacity for the hepatic phase or, I mean, from what I've read of it, the intrahepatic phase participates to a considerable extent in the endocytic cycle. And there are mutations, the human diseases that affect that cycle. Is there any evidence that that plays a part in malaria?
We would have to sequence the genomes of all these different apes to see whether there are similar changes in their genes, and we haven't done that.
Okay, well, listen, thank you immensely, and we will proceed now with Caroline. And then
We should get this one. This one first. Oh.
Hello? Hello? Yes? This is forward. And so the time is like this. Okay, good afternoon. So we're going to switch gears a little bit. Our lab is interested in understanding the interactions between Plasmodium falciparum and the mosquito immune system that affect malaria transmission. So I would like to start with just a couple of slides telling you a little bit about what is it about malaria infection that makes humans so sick and causes so much disease. Just I will briefly touch on that. Then most of my talk will be, I will be trying to give you a sense of what happens when a mosquito gets infected with malaria, how the mosquito immune system tries to contain this infection, and the evidence that there are very powerful antiplasmodial responses in the mosquito that can limit infection. And then, of course, if the mosquitoes have such a great immune system, then why is malaria transmission so high? This is something that has always puzzled me. And I think the answer is that Plasmodium falciparum is not a normal parasite. It's a parasite with a trick. It's a parasite that has learned to make itself invisible to the mosquito immune system. And I'm going to tell you the evidence we have of this and the genes and the mechanisms that make this possible. So, as we mentioned before, malaria is a very severe disease that affects 40% of the world's population. 90% of cases take place in sub-Saharan Africa. And I'm talking about falciparum malaria in this case. So humans get infected when the mosquito injects sporozoites. They infect the liver, multiply, and then they go through cycles of infection and multiplication in red blood cells. And basically, most of the severe disease and damage to the human comes from two things. One, of course, is anemia, because you have constantly bursting red blood cells. So that's part of it, chronic anemia. But the severe disease, the life-threatening, is because erythrocytes that are infected express parasite proteins on their surface that make them sticky. It's like they have a Velcro. And the parasite does this because if it did not site right here, it will be the infected red blood cells will be destroyed by the spleen and the parasite will be eliminated. So the parasite can hide from this clearance mechanism by attaching to endothelial cells. So if it attaches in the endothelial cells of the brain, then it causes severe brain malaria that can result in death and coma. And in other children that do not die, it can have also long-term consequences in terms of brain damage. Usually by the time females are in an age to have children, they are resistant. I mean, they have antibodies to malaria. They've had many cases. But when the woman becomes pregnant, then the placenta creates a new place where the parasites can hide. So parasites with particular forms of these adherent proteins now can hide in the placenta and then can cause severe malaria in this person. And also, this binding of the erythrocytes to the placenta disrupts the flow of blood in the placenta, and this damages the fetus. And this has severe consequences. It's estimated, even today, after the introduction of bed nets and all of these, that every 45 seconds a child dies of malaria. And every year, an estimated 10,000 women and 200,000 of their infants die as a consequence of placental disruption, malaria during pregnancy. So, sorry, I'm trying to go back, and I think I keep going forward. No? Yeah, sorry. There we go. Sorry. So, I won't move my finger. I have to glue it. So, I think something that is, of course, obvious is that the disease in humans starts with the bite of a mosquito. And I think nowadays everyone accepts that everywhere in the world where malaria has been eradicated, this has been achieved by controlling the vector. And this is because mosquitoes are extremely efficient. To give you an idea, if you live in an endemic area of Africa, you will get a 400% coverage, meaning everyone in the place will get infected with malaria 4 times every year. So with that rate of reinfection, no matter how good the drugs are, no matter what measurements you take, it's just not possible to control. And this is also what makes a vaccine so difficult, because it's extremely, extremely efficient. We work with the mosquito Anopheles gambiae because this is a major vector in Africa. And this mosquito is remarkably efficient transmitting malaria. And this is one of the reasons why malaria is such a big problem in Africa. Much more severe than in other places, because transmission is much more efficient here. So now in my talk, I'm going to spend a little bit of time in these first 2 parts, basically giving you a model, what we have learned, using mostly Plasmodium burgea, a mouse model of malaria, as to what happens to a mosquito when a parasite invades the midgut, how the midgut cell tries to contain the infection, the epithelial responses, and then how these epithelial responses also affect the response of the complement system. So the mosquito has a complement-like immune system that can bind to the parasite of the surface of the parasite and trigger the formation of a complex that will lyse the parasite. And it's very powerful. In some cases, it can completely block transmission when it's effective. Then I'm going to talk a little bit about signaling transduction pathways and how they can regulate some of these. This is going to be very briefly. And I would like to spend most of my time in this part, and it's showing you the evidence we have that the Plasmodium falciparum has mechanisms that make the immune system of the mosquito fail, and that when this control of the infection in the mosquito fails is when you have really high transmission of malaria. So the mosquito, most of this part I'm going to talk about is in Plasmodium burgea, but it applies also to other malaria. So mosquitoes get infected when they take gametocytes, blood from a host that contains gametocytes. Sexual maturation and fertilization takes place. They form a motile ookinete. This ookinete is a banana-shaped parasite that moves. You should see the videos. It's amazing. It just crawls and is able to invade the midgut cell, doesn't form a vacuole, it just inserts itself through the cell and causes irreparable damage to the invaded cell. But the cell dies, but putting a fight until the end. The cell will respond to this invasion by triggering some production of some chemicals that can be toxic, nitrogen, nitration reactions, and the parasite has to withstand these responses, and as it emerges from the cell, then it has to face the complement system of the mosquito. There are soluble proteins that are produced in the hemolymph, which is like the serum in the circulatory system of insects, and this protein can bind to the surface of the parasite, and a very famous member of this family, TEP1, that has been very well studied, is similar to C3 factor of complement. It will bind to the surface of the parasite and form a complex that will lyse the parasite with other proteins. All the components are not known yet. Now, something that was very puzzling is that you can infect a mosquito with a mouse parasite that is clonal. So genetically, all the parasites are identical. Then you see that TEP1 binds to the surface of some of these parasites, but other ones are spared. If the TEP1 binds, this parasite will die, but then these ones will survive. But how come it binds to some and not others if they're genetically identical? You can sequence them and they're identical. So why? And the picture that is emerging is that although it's step 1, the one that executes the lysis of the parasite, the fate of all kinates at this stage of the parasite is determined during their crossing of the midgut. So basically, whether a parasite will survive or not depends on how well it fends the epithelial response to the invasion. So I will show you some of the data, because I cannot review it all, but I give you some feeling of the kind of experiments we can do to study this. So these are some pictures where you can see when the parasite invades the midgut cell, it induces the expression of nitric oxide synthase. Here's in green. You can see that the invaded cells are staying stronger. This is a surface parasite protein. You see the cells butt off into the midgut lumen and are going to be eventually expelled from the epithelium as the neighboring cells come together and the invaded cells are repaired. The cells also not only express high levels of NOS, but also heme peroxidase. We have identified this gene, and this is interesting because in human macrophages, it has been shown that you can have NOS starting a nitration response, but you can have a second step that involves peroxidases that can greatly potentiate the efficiency of these reactions. So we had the hypothesis that this was also happening in mosquitoes. So we identify the peroxidase that is mediating these responses. And I will show you some of the data. This is all published, but I will just summarize here. So for instance, we see that this peroxidase accumulates in the cell that hasn't been invaded by the parasite. We can measure midgut nitration using an ELISA-like assay using anti-nitrate tyrosine monoclonal antibodies. And we see if you compare the level of nitration in a control versus an infected midgut, Plasmodium infection induces more nitration. But if you silence this peroxidase, you reduce nitration. And this is a control that was infected with LAC. This is a gene not present in the mosquito. But if you inject double-stranded RNA to this peroxidase, you can silence the gene. And if you reduce expression of this enzyme, you get reduced nitration. When there's less nitration in the midgut, you have higher parasite survival. This is, for instance, this is a group of mosquitoes, 41 mosquitoes. And this is the number of parasites in each mosquito is a DARC. And you can see that the median level of infection increases significantly when you silence expression of this peroxidase. So this is to give you a feeling for this. This peroxidase also works with another enzyme, NOx5, that provides hydrogen peroxide for this reaction. And we found some genetic evidence that there was a link between this chemical nitration and TEP1 activation. So we tried to find some direct evidence. And here is a similar picture to what I show you. This is parasites that just finished invading the mosquito midgut. Some of them are covered with TEP1, here in green, and some are not. So this is the number of parasites staining a control, and this is when you silence the peroxidase. And you can quantitate this in two different ways. One is you look at percent of TEP1 positive parasites, and you see that the number of parasites is reduced significantly if you silence the peroxidase. And we can also measure TEP1 binding to the parasite with ELISA, and it's greatly reduced. So what this is telling us is that there is a connection between the midgut response and the ability of the complement to kill. So I would like to take a minute, and I think in images. So the story at some point gets a little bit complicated, but I think if we have some images, it's going to be easier to follow. So that's a mosquito that just took a blood meal. The blood meal was infected. So let's look inside. So this is the sexual forms of the parasite. Fertilization will take place. An ookinete is formed. This ookinete then has to cross the barriers that normally separate the blood meal from the epithelium, crosses the peritrophic matrix. This is a chitinous membrane that prevents bacteria from coming normally in direct contact with the epithelium. Then, as these barriers are broken, then the microbiota comes in contact with the cells. The parasite invades a midgut cell. And now let's see how this cell will respond. So the parasite invades the cell, and then, as I show you, this triggers the expression of nitric oxide synthase, right? And nitric oxide produces NO. NO is very reactive, right? But because it's so reactive, it decays quickly into nitrite. It cannot really accumulate in the cell. So if you really want to have a strong reaction, you have to accumulate some of this nitrite. And then you can do a second step, where if you have a peroxidase and an enzyme that can give you some hydrogen peroxide, if you have these components together, then you can do a second step and produce nitrogen dioxide. which is a second chemical that is very reactive. Now, if the parasite is exposed to this chemical, then the parasite is modified, is changed somehow. We still don't know what is the nature of that modification. So if this parasite has had this exposure, then TEP1 will bind to its surface, and this will trigger the formation of the complex that will lyse the parasite, right? So this is a case where the immune system was successful and transmission did not happen. But if you have invasion, you have the induction of NOs that is very fast after invasion, you can produce NO and you have nitrite. But if you can block the second step, the potentiation step, and in the lab we can do it by silencing expression of either NOx5 or HPX2, then the parasite will come out, but the potentiation didn't happen, so nitration is not effective. So this parasite then will be there, but TEP1 will not see it. So then this parasite that is not visible then can go ahead and develop, multiply, release thousands of sporozoites eventually into the hemolymph that will go into the salivary gland of the mosquito and be transmitted to when the mosquito bites the next person, right? So let's talk a little bit about signal transduction, right? And this is going to be very brief. I just want to mention that we have evidence that the JNK pathway has a critical role in antiplasmodial immunity. So in Drosophila, and in humans, the JNK pathway is involved in multiple responses, of course, including the immune system. In Drosophila, it's involved in development, immunity, apoptosis, and stress responses. And I'm showing here some of the genes in the pathway. The ones showing with these geometrical forms are orthologs of these genes in the mosquito, so that they're present in humans and in the mosquito, and in Drosophila. And then if you silence expression of the genes required to activate this pathway, if this pathway is important to kill the parasites, if the pathway cannot be turned on, then the parasites will do better, right? You can also silence this suppressor, which normally is the brakes of the system. It turns off this pathway. So if you silence this, you can overactivate the pathway. So it's like a car where you remove the brakes, right? So it's going to be overactivated. So what happens? Just to give you a feeling, this is two examples, two genes required to activate the pathway. If you silence them, the parasites do better. This is telling us that this pathway is important to kill parasites. If we overactivate the pathway, we have the opposite effect. Now the parasites do worse than when this pathway is not active. So we also did a series of studies trying to understand how this effect on antiplasmodial immunity was working. And to make a long story short, we found that the JNK pathway induces the expression of NOx5 and HPX2, and these are required to have good nitration. If this pathway is not induced, then nitration is ineffective, and then you get much more parasites survive. So, okay, I would like to take a minute here and just summarize what I've said so far, because it's going to be critical for the next part. So I want to invite you to imagine that you're sitting inside a midgut cell, right? So we're all inside a midgut cell, and a giant ookinete is going to walk through this door. Giant ookinete is going to enter. As soon as it enters, an alarm will set off, right? This alarm will tell the system that this is a forbidden zone. This is a barrier zone. Nothing should cross this. Now, when this happens, then the alarm is set off, and then a chemical will be produced inside the room. This chemical is not strong enough to kill the parasite. So we can think of it like a fluorescent paint. So imagine the ookinete is passing through this cell, and a fluorescent paint will be made that will tag the parasite, right? When this parasite crosses the cell and gets out, it's covered in fluorescent paint. So it will be immediately recognized by the security system, and the parasite will be taken. Now, if the parasite manages to cross this room without being labeled, so that, for instance, by disconnecting the alarm, if the JNK pathway is not active, there is no alarm, there's no production of paint, parasite can go through, gets out, and the security system doesn't detect it. It's as if the parasite is not there, right? Does anyone have questions so far up to here? Okay, so now we're going to see what happens. To give you an idea of how strong the TEP1 system can be, this is infection in a normal mosquito. If you silence TEP1, you increase 4 to 5-fold the number of parasites. So basically, this system normally kills about 80% of the parasites in this model, right? Now, people were all excited about TEP1, and then people start trying it with Plasmodium falciparum, and this is what happened. You infect, this is an African, a line of African origin with an African mosquito. You silence TEP1 and nothing happened. And this shocked our community. One interpretation is we had lost 15 years of our life barking off the wrong tree, and all these models are irrelevant to humans. That's one interpretation. But that is not the case, luckily. What I hope to convince you is that we needed to see these to know that how the system could work. Now, the question is, why is it not working here? Is this normal, or is this something unusual about this parasite? So we decided to look at this in more detail, and I will show you the evidence we have that Plasmodium falciparum is able to evade the mosquito immune system. So this started with a line that was selected at NIH about more than 20 years ago, almost 25 years ago. And this is the refractory line. In this line, the parasites develop, they invade the midgut, but as soon as they come in contact with the hemolymph and with TEP1, then the parasites are damaged. But in this case, when they are killed, they are immediately covered with a black pigment. And so they cover this black pigment is called melanin, so the dead parasites are melanized. And this is nice because we can see when a parasite has invaded but died. And this will be is very useful to us. So when we infect this strain was initially selected to kill all Plasmodium cynomolgi parasites. But then they started testing other parasites once the refractory strain was available, and it kills all the parasites that you can imagine, with one exception. If you infect this line with Plasmodium falciparum from Africa, from where this mosquito comes from, then it does really well. So that began to say there's something about where a parasite strain comes from and whether a mosquito will see it or not see it. So to give you an idea of how drastic some of these differences are, this is an African GB4 line from Ghana. And in these experiments, I'm going to be showing you each dot again is the number of parasites in a given mosquito. And in the x-axis, I have the dead parasites, the number of melanized parasites, y-axis, the number of live parasites. So in this one, in the African melanization, we never saw melanized parasites. All of them were alive, right? While if you infect with a Brazilian strain, then now basically almost everybody, 97% are dead, and basically almost no one is alive. So the way this mosquito responds, and these are both Plasmodium falciparum, but depends where the patient infection came from, right? So this told us that there was something different. Now, because these two are Plasmodium falciparum, then you can cross these parasites, so you can do a genetic cross. And a cross between these two lines had already been done at NIH to map erythrocyte invasion phenotype in Tom Whelham's lab. And we wanted to know, I mean, before we went into mapping, we decided to know why are these Brazilian parasites dying, right? So they die. And if you infect a mosquito that has been injected with a control like C, of course they also die. But if you silence TEP1 so that the mosquito immune system is no longer active, now the parasites survive very well. 97% of them survive. So the Brazilian parasite is killed because the mosquito immune system can actively trigger killing, right? Now, when you silence TEP1 in the African strain, what you see is that the median of infection is exactly the same. The number of melanizations decreases from 4% to nothing, but other than that, this is telling us that 96% of the parasites already can escape the immune system. So this is a lousy response. This is a very effective immune response. So why is the mosquito responding so different to these two parasites? So we did use a combination of a classic QTL from a genetic cross between this African and Brazilian strain, and in combination with I'm blanking out, a group linkage selection, sorry, with group linkage selection. And then using a combination of these two, we were able to map a locus in chromosome 13 that codes for 41 genes. Then using knockout lines, we were able to determine that it is PFS47, the one that allows the parasite to evade the immune system. And I will show you the data that support that in a minute. Now, this locus turned out to be very interesting because right next door to PFS47 is another gene called PFS4845. This is a gene expressing male gametes, and is a major top candidate for transmission blocking vaccines. Also, very close to here, there is a protein called Kelch protein 13, which is a marker that is strongly associated with artemisinin resistance. So, and this will become important at the end when I see what is the relevance of this association. Okay, so if the parasite has the PFS47 gene, as I told you, most of them do well. In this case, 93% survive. But if you remove this gene, so you have a PFS47 knockout, now the African parasite, this is NF54, they die. So if you remove this single gene, PFS47, from the African parasite, it can no longer survive in this strain. Now, if you remove the gene in the mosquito, in the parasite, right, so that now it's dying. But if you infect a mosquito that is wild type, so it's infected with LAC-C, it dies. But if you disrupt the mosquito immune system, now the parasites survive even if they don't have this gene. So this is telling us that the gene is only required to avoid the mosquito immune system, right? That the parasite doesn't need this gene to survive per se, but it's just to be able to not be killed by the mosquito, right? So, and this is, we also tested the same gene, whether this gene was also necessary in other strains. This is the wild type. This is a mosquito that is susceptible and doesn't melanize the parasite, but if you, susceptible, but if you infect, this is another strain, the S strain, so this is different from the refractory strain. When you don't have PFS47, then you have very low infection. So even in strains that are non-melanizing, so some mosquitoes kill the parasites and lyse them, and some kill and melanize. This is a different genetic background of mosquito, but you also need this gene because you will get very little infection, but if you silence the complement now, you can get high levels of infection, right? So I didn't mention it here, but we also showed that if you take the knockout line and you complement with the African allele, you can rescue the phenotype. But if you complement with the Brazilian allele, you do not rescue for the refractories. So we also wanted to know, PFS47 was known to be expressed on the surface of female gametes, but because the ookinete is the stage that has to face the immune system, it was important to show whether it's also expressed. So we find that PFS47 is also in the presence of wild-type parasites here, but it's not expressed on the surface of the knockout parasites. And when we look at we ask, how does the mosquito midgut respond to a wild-type versus a knockout parasite? Is there a difference in how the midgut is responding? What we find is that the wild-type parasite that has PFS47, there is very little nitration. Actually, the level of nitration in infected midguts is less than those fed in uninfected blood. The opposite is true for the knockout. When you have the knockout, the nitration is much higher in the infected than in the control. Also, we found that if you have PFS47, you no longer induce expression of NOX5 or of HPX2. So our current working hypothesis is that falciparum is evading the immune system by disrupting somehow J&K signaling. And we're doing a series of experiments to demonstrate that this is the case. So basically, if the parasite has the right allele of PFS47, it can invade the midgut, but it inhibits induction of the potentiation step of nitration, then these parasites are going to be invisible, develop, and go on to transmit. If the parasite has the wrong allele, for example, the Brazilian allele in the refractory strain, or if it lacks PFS47, then it will be modified by activation of these enzymes, and then the TEP1 system will bind to its surface, trigger lysis, and then this parasite will die and not go on to transmit. Now, there's something very interesting about PFS47, so we're going to go from the lab to the real world, right? We're going to make a big jump. So if these are just barcodes just to look at different haplotypes, right, of PFS47 or 48, 45 in a collection that we have at NIAID. And I left it like this because so each different barcode has a different color. Basically, as malaria originated in Africa, so you get more diversity in Africa. This is expected. But you see something interesting in this sample size. There is one haplotype that is very common in the Americas, the only one that we find. In Asia, there is a dominant one. And there are some haplotypes in Africa that you don't see outside of Africa. And these had already been noted by Conway. For example, he found, this is in a small sample size, you have, in this case, 3 haplotypes of PFS4845, but when you go out of Africa, you find 2 of them in Asia and then 2 different ones in the New World. Now, it has been hard to explain why do you get geographic structuring of this kind of gene. And what we would like to propose is that is the vector. That it is the geographic structure is determined by selection by mosquito vectors. And to this, I think we need to take a minute to look at a little bit of the history of anophelines in that transmit malaria. So just to simplify this is the world map, and these are all the anopheles species that transmit malaria in different places. So just the overview, I want you to realize that there's many different colors, meaning there's many different vectors in different regions. So mosquitoes are local, right? The second thing is that, malaria originated in Africa, and this line highlights the area where Anopheles gambiae is the main vector. But these mosquitoes and those from South America separated from each other a long time ago, 95 million years ago. So since the continents separated, these 2 mosquito species have not seen each other, meaning at that time they were still biting dinosaurs, right? Our ancestors were rats, right, underground, hiding from the dinosaurs. They had not gone extinct yet. So it's been a long, long time since mosquitoes from South America and from Africa have seen each other. So now, suddenly, humans begin to walk out of Africa, and then they begin to move. And if a parasite wants to be transmitted, it has to adapt to a new vector, a local vector. Those parasites that can adapt will be transmitted, and the ones that cannot make the jump are in a dead end, and infection will not be spread in the new place where this infected person comes. So now we have analyzed in more detail haplotypes of PFS47 in human. This is from human samples. This is 178 isolates, and we found 30 different haplotypes. And you see that there's much more diversity in Africa, and this is expected because this is the origin. If you look in Asia, you see that the alleles that are very abundant in Asia are different from the ones in Africa. In Papua New Guinea, you get different ones. And the most extreme case is the Americas, where so far we have found a single allele of this gene, an allele that is rare in Africa. So how do we explain this? What we think happened is that mosquitoes were being transmitted here in Africa, very well adapted to their vectors, and suddenly, historically, the New World is discovered, and with the slave trade, it's estimated that more than 7 million Africans, many of them, or most of them, infected with falciparum, were brought up into the New World. And this happened within the last 500 years, that in evolutionary terms is a very recent event. So now, these parasites find themselves in humans, in places where the mosquitoes are like from another planet, right? They're really, really different from the ones they are used to. So we think that only very few, actually only one haplotype that we can identify so far was able to make the jump. And it's only these parasites, the one that have been transmitted in the Americas, right? So this is, of course, a hypothesis, but can we find experimental evidence to begin to support this hypothesis? So if you take, for instance, now, I just want to call your attention to Anopheles albimenus here in yellow. This is a vector in the Caribbean, South America, in Central America, and a little bit in Colombia, mostly in the coast, in the Atlantic coast. So if you look at Anopheles albimenus, so this is a New World vector, right? If you're infected with an African NF54, the infection is very low. We're using Anopheles stephensii here as a positive control. This is a strain that has been selected to be very permissive to infection with all falciparum. So it always infects with everything. So it's our positive control that the infection was good. So if you take another strain from Senegal, the same story. But if you take a strain that came from Brazil, now it does really well in the New World vector, right? Now, when these infections don't happen, we can ask, okay, so why is albimenus not being infected? Is it that it's just incompatible? Maybe there's a difference in biology, or is really the immune system? So we have been able to identify a key component. LRIN1 is a key component of the complement in this species, in Anopheles albimenus. So you can take the African parasite. If you disrupt the mosquito immune system, now you get really good transmission, infection with albimanus. So what this is telling us is that this is giving evidence that it is the mosquito immune system, the one that is limiting transmission of certain haplotypes. So an African haplotype was never able to make the jump into the New World because the mosquito immune system is very able to see these parasites and eliminate them. That's our thinking, right? So right now we are constructing a series of lines where we are making the PFS47 jamboree. We're taking one parasite, NA54, and we're changing alleles from Africa, from the Americas, and now we're infecting vectors in the collection at NIH that come from all regions of the world, trying to see what is the language of compatibility. And we think that there are matches and mismatches. In this case, we think this is the most extreme situation, but we think there's almost a yes or no answer, but we think there's going to be shades of gray here. And this is important because if we understand what is the language of compatibility, we can predict what is the chance that a certain parasite will be transmitted. For example, I talk about the Kelch protein that is associated with artemisinin resistance in Cambodia. So if you have this resistance, what is the chance that this parasite line, for instance, will come into the New World and spread? Based on this, what we know so far, very little, because those parasites never were able to establish in the New World. Now, if these parasites, can they jump back to Africa? I think yes. So I think that as we understand the molecular basis for this compatibility, it can really give you very strong epidemiological tools to predict which parasites are likely to spread in an area and understand what makes a compatibility or not. So, of course, many questions still arise, and we're trying to understand how this works. For example, if there is a PFS47 that is polymorphic, there has to be a polymorphic receptor in the mosquito that is the counterpart that PFS47 is interacting with. We're working hard trying to identify it. Also, if having PFS47 is a key to the very successful spread of falciparum malaria, could that be a weak point in the system? Could that be an Achilles heel where if we had a transmission blocking vaccine and now we make these parasites visible, would mosquitoes eliminate them and not transmit them anymore? So this is where we are working. And of course, our long-term goal is to try to disrupt transmission to prevent human disease and improve the quality of life of those living in endemic areas. I just want to briefly introduce some of the people that have done this work. Giselle Almeida was the person heading the project where we found the connection between epithelial nitration and TEP1 activation. Lindsay Garber did the work with the JNK pathway and the importance of this pathway in anti-plasmodial immunity. Alvaro Molina Cruz is a staff scientist in the lab, and he had the mapping team. This was a 7-year project to map this gene, very challenging, but at the end, I think it was all worth it. And then these are the people from the lab that are working very hard trying to understand how this works. And also some new collaborators. Oliver Bilker from the Sanger Institute, we have introduced PFS47 in P. burgii, and we are asking, is it enough to have this gene for a mouse parasite to also become invisible? Is that enough? And within the infection, we should have the dissections tomorrow afternoon. So we're hoping that that would work. I think will be an amazing tool. Martin Boulanger is helping us express this protein to solve the crystal structure and try to understand how it works. And we're doing field work with Isabel Morley in Cameroon, with Napomon and Mali team and MRTC, and with Pablo Pimenta as part of a Science Without Borders. We're also analyzing, we just established an Anopheles aquasalis colony, and we're doing the compatibility. And I would like to stop here and thank you for your attention. Yes.
So Carolina,
Yes.
when the slave ships arrived,
Yes.
the mosquito was the wrong time.
Yes.
They all came with various different versions of falciparum.
Yes.
Was it just luck that one of the many, many, many that arrived in the ship happened to fit to fly under the radar?
This is what we think, that there was one, and actually it's a rare one. We only have one sample in all these collections. Of 178, we only have one with the New World allele that came from Africa. But this is, I think, what happened, that it was almost, there was a high chance that it wouldn't have happened, right? It's just that there was one parasite that had just the right combination. So what we're doing now it's difficult to compare because when you compare lines of different continents, they have many differences in many genes. So what we're doing now is we're making isogenic lines by having just all of them with the same genetic background and just change this gene and then try to dissect the compatibility question with each of the different strains. But so now we're in the process of cloning a line that is African parasite, but has the New World allele expressed with the right regulation, everything right. And our hope is that this parasite now will jump into the New World for the first time. So having that jump, we think might be enough. But we have a preliminary with plasmids transformation, but those experiments are not very clean because of dosage problems and multiple drugs that have to be kept. So we think that the transformants are going to give us a clean answer.
So, Carolina, what's the effect of global warming going to be on the distribution of mosquitoes?
Well, I think that, I think the distribution will change. So places where you did not have, let's say, compatible combinations may change. So you may begin to see transmission in places where you have not seen them before. I think it's more worrisome for mosquitoes like Aedes aegypti that transmit dengue and other diseases that are less specific. I think falciparum in general, mosquitoes tend to be local, and I think they might spread. I think the biggest example of how important the vector is, in the 1950s, it was arabiensis, right, Albert? Some Anopheles arabiensis, which is very similar, it's a sibling species close to Gambi, it's part of the Gambi complex, was brought into Brazil by mistake, and they began to have epidemics and very high mortality in Brazil. Luckily, someone noticed and realized what was going on, and they did a campaign of eradicating this mosquito from Brazil, and they were successful. If they had not achieved this eradication, South America will be like Africa today, I think, in levels of transmission. So I think it's the vector. I mean, the vector is really critical. I think the severity depends on how many the infectious potential of a given mosquito, and that for Gambi is, like say, the maximum, because this mosquito basically doesn't see the parasite, so allows all the parasites to go through. There's no filter, basically, in this case.
We bring back DDT?
Well, I think under certain conditions, some people think that it could be useful, but of course, mosquitoes develop very quickly resistance to DDT. So I think massive spraying like it was done before with the idea to eradicate, I don't think so. But a part of an integrated campaign with long-lasting DDT paint to reduce mosquitoes when you are introducing vaccines or other measurements, I think it has a place under the right circumstances. I think we can.
Yes.
I read a recent article about Oxitec in Brazil, and their modified mosquito. I guess, do you have any opinions or
yes,
about that?
That's a very interesting system, but this is for Aedes aegypti. But I think it's a good point. So Oxitec, what they have is a system called Riddle. So these are mosquitoes that the males have a gene that when they mate, then the females of the next generation will be unable to fly. So it's a mosquito that just will be paralyzed and not. So if you release a lot of these males and they mate with the local population, they'll find the females, and then the mosquito population will collapse. So I think this could be very useful, for instance, if you're about to have a dengue hemorrhagic fever in Rio de Janeiro, and you have to stop those mosquitoes and you have to do it quickly, I think could be a very good measurement. The problem is that it's not a long-term solution. Basically, you have to release thousands of mosquitoes every year, so it ends up being very expensive, right? So I think it has an application, but by itself, I think it's non-sustainable because it's very expensive. Now, there's some exciting things for Wolbachia, which I don't know if you want to discuss.
So this might be a stupid question, but do you think that the PFS47 is causing a surface protein or is inhibiting a surface protein on the parasite, which doesn't allow for it to be marked, like either it's inhibiting or?
This is the million-dollar question. So the short answer is we're not sure how it's working, but we know that we can make recombinant PFS47. So we take the critical, what we think is the critical part of PFS47, and if we incubate cells in vitro with these, the cells no longer activate the J&K pathway. So we think that it can interact with some receptor, and we're very several people in the lab are working very hard trying to find what is this receptor in the mosquito. So it is binding to something, probably on the surface. We are not sure, but the cell line experiments will suggest that it's on the surface. And somehow this cell no longer triggers the alarm. So in this image that I was giving, imagine that someone disconnects the alarm in the door. So then the parasite can get in, but it really somehow the system is inhibited. We know there is no nitration, no induction of NOx5 HPX2, and that putting these extracellularly can block signaling. Now, this is in a cell line assay where we activate J&K by interacting with heat-kill bacteria in a cell line, but we see lack of induction of the effector genes when we do that. So we're doing it because it's simpler. You don't have invasion, you don't have injury, because this is a complex system, because the cell is undergoing apoptosis too. And something I didn't talk about, but also in this picture, which I think is going to be central players, mosquitoes have white blood cell like hemocytes, which are leukocyte-like cells that we think are central in this. And I did not even bring them into the picture because it gets mind-boggling at some point. But I think they are going to also be major players. So I think there's interactions with all of them that at the end decide the outcome of whether a mosquito will make it or not.
Are there efforts to try and interfere? If you could block the hepatic phase,
yes,
you'd stop the cycle.
Yes.
Now, we seem to be learning a lot in recent years from the basic cell biology of how hepatocyte vacuoles
so
form,
I would say that there are
acidification and all that.
Yeah, I would say that there are three types of vaccines that are underway. One is transmission blocking, which is like a person that is infected will no longer transmit. Like if you get malaria and a mosquito bites you, you cannot infect mosquitoes. That is good at an epidemiological level, but it doesn't benefit the person who received the vaccine, right? Now, people have pre-erythrocyte vaccines, which are stages before the erythrocyte, so hepatic stages, and there are many groups working on that. The problem is you have a very narrow window, right? From the time the mosquito bites to when the parasite goes into the liver, you have a few minutes, basically to maybe hours. So the vaccine has to work very quickly, right? Once it goes into the liver, one parasite multiplied, and then once it goes out, so just to give you an idea, in a mosquito, you get 1 or 2 oocysts surviving. That's the normal level of infection. In a human, once you go to the erythrocyte stage, you get 10 to the 11 parasites per person. So the numbers are staggering. Once you have that many parasites, then it becomes very difficult to control. So other people think that maybe it's okay to allow the infection to happen, and we should start maybe preventing cerebral malaria first. So try to develop vaccines that will prevent cerebral malaria. Because it is, once you get out of the liver, it is hard. Now, there's some new promising asexual stage vaccines that we appear to understand better what is the right conformation and the right way to present some antigens. So Lou Miller has some very interesting results with surface proteins required for invasion, so that they can be very good antigens and you can prevent erythrocyte stages.
How does vivax deal with the alarm system?
That's a great question. So we have just started to work with that. Now, that's much more difficult because there's no in vitro system. There's no way to study vivax. But what we're about to start doing is in the new world, we've only found one allele of vivax. So now we're trying to put this, we're thinking of putting the vivax PFS47 into the falciparum knockout and see if that's enough to open the doors into the mosquito, right? My guess is that vivax may be more able to jump to different vectors, but I don't know. I don't know, Albert, if you have any speculation, right? With vivax, we don't know. Yeah. So basically, we have some samples, but our pool of PV samples is small at the moment. But so far, all the vivax that we have sequenced, and we have quite a few samples from Brazil, there's only one allele so far that we can find. And it's the same allele that has been reported in El Salvador and Honduras. So maybe also there's only one allele, but we don't know yet.
We have all sorts of theories that are now becoming more out in the open about the origin of viruses. What do you think is the origin of a Plasmodium?
The origin.
Origin.
I think that mosquitoes maybe would get infected. They say that mosquitoes used to suck on plants first, so like they will take sap from plants, and there's some parasites that infect some insects that take them. Now, what is the jump? The thing is, what is first, the chicken and the egg, right? So what was first? Was the mosquito first infected and then it adapted to humans? Or did humans have the infection and then it jumped into the mosquito? It is hard to imagine because you need both to have any transmission. So how was the cycle first? I don't know, Alvaro, if you have any.
Well, one of the, this is total speculation, but one of the theories that some people have proposed is because the part of the malaria cycle, life cycle, that has the sexual stages and is more complex is the one that is in the mosquito. Probably that phase was first. And the parasite also has the picoplast, which is an ancient chloroplast organelle from an aquatic organism, most likely, and the mosquito has an aquatic stage. So probably the first Plasmodium was a parasite in the aquatic stage of the mosquito, the larvae. And maybe later on came the accidental host, the human.
And mostly, probably the mosquitoes were feeding on plants or plant nectar, plant, some kind of, because there are insects that take the sap of plants, and I think the mouth parts, all of that, started like that. And then someone bit a human and they got a super meal. So they said, oh, wow, this is good. So then they multiply, and that's how it started. Right?
Are there any other questions? Well, I would just, two things. One, I'm reminded, I think, that there have been 4, malaria has been the greatest subject for numbers of Nobel Prizes, I think, of certainly any infectious disease. I believe there were 4 Nobel Prizes given, 3 of which were really appropriate. One of them, fever therapy, has sort of died by the wayside. But you can see that from this incredible explosion of information and how we're sort of, you get your feeling, I got the feeling today that we were very privileged to be on the crest of all kinds of waves that are coming, and we can do nothing but admire the quality of your work and your presentation, and thank you very, very much for being here this day.
Thank you.